So as an example, we've set up this idea of having some understanding of a distribution of outcomes for us to be able to do a statistical analysis. Now, this is a very simple, these normal distributions, whether they're flat or very pronounced, are fairly simple distributions. And for some populations, that works really, really well. If we're looking at height in humans, follows a fairly normal distribution. That's a fairly simple thing to analyze. Now, when we are dealing with phylogenies, our probability distributions are much, much more complex. And this is because phylogenies are complex, and there are so many of them. And so, although it's not ideal, maybe the best way to think about it is like a three-dimensional, but in reality it's multi-dimensional space. And so in this, we might have some areas that are pretty good. These local peaks, and the higher we are here, the better the tree scores are. But this is a very rugged and difficult topology. And so because phylogenies can have such a complex probability of outcomes, because we don't know what that might be ahead of time, we need a way that we can estimate and look at this probability outcome, this search space. And to do that, we use a process called Markov chain Monte Carlo. Now, you don't need to know all the details about it. It's a fairly advanced statistical approach. We're going to give you the bird's eye overview. So it's a way to quickly simulate and estimate what the probability distribution is. Okay? And to do this, very simply, what you do is you pick a random point to start at. And then you use some sort of an algorithm to sample areas that are nearby. And I'm going to use the analogy of a landscape. Let's say that you're looking to find the highest peak. You're optimizing elevation, and you're trying to find the highest peak. So what you do is you randomly drop an individual off, and then because you can't look around, right? So in a phylogeny, you've got an individual phylogeny. You can't just look at the phylogeny, look at all of the phylogenies next to it and see which ones are better. You have to test them. And so in this analogy, let's say you have a blind individual, and you're trying to help that blind individual find the highest peak in an area. So you drop them off, you have them do some explorations right next to them. So they take a step north, south, east, west. And if one of those steps makes them a little bit higher in elevation, that's the new place. And then they go from that one. They do north, south, east, west, and that's the new place. And so by doing a very quick, rough algorithm, we could potentially continually go up in elevation until we reach the highest point. At that point, they would keep going north, south, west, get lower, and we would sample this area and sample and sample and sample. That would be a local maximum. It's no guarantee that it's overall the global maximum. And so we would randomly start another one. And maybe we randomly start here and we go up, 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 and then we sample this peak. And so this is a very fast method for sampling very rugged search topologies. And we're using this three-dimensional search topology as an analogy. But in reality, remember that phylogenetic search space is a multi-dimensional space, and a very, very large one that we might need to spend some time and effort sampling. But our Markov chain Monte Carlo is an approach to sample the probability distribution and do so in a way that is fairly certain of guaranteeing finding us all of the really best ones, whether it's a very rugged space like this where we have a few very good answers, or it's a little bit more spread out and we'd have a sampling distribution that was a little bit more spread out. And so this is, again, as long as you know the details, which is what we pick a random start, we look around it. If we find a better answer nearby, that becomes our new one. We do that over and over and over again until we've sampled the search topology fairly rapidly. Okay. Now, in addition to this Markov chain Monte Carlo for sampling the probability search space, we then use those probabilities to calculate a model of evolution. And what we see when we do this for most analyses is that we start off with a model of evolution and we find a tree. We then allow that tree, using this Markov chain Monte Carlo search strategy, we allow that tree to then inform, that becomes our posterior probability, and it informs our model of evolution, we re-estimate the parameters for that model of evolution, and then we run the analysis again. And we do that over and over and over again. And what we find is that when we start randomly, we usually don't have a really great score. But our scores usually will improve rather rapidly until we are getting really similar scores. And as we do this over and over again, right, allowing our posterior probability to then inform the parameters for our next round of that analysis, and we get scores out, and we usually will find a plateau. And this plateau represents a peak in scores where we found a very best overall solution. And if it's a very rugged search topology, we might get slightly different trees during another random event, but the scores are going to be very similar. If it's a very, I'm going to Google, I'll bring in some scores, some possibles over here. I grew up in an area in Arizona where the nearby mountains were called the Superstition Mountains. And I choose them as an example because they have this very rugged search topology. If you're trying to find the highest peak in the superstitions, it could be difficult. And this is just the most famous set of peaks, but there are actually others. So here, let's use this one. Let's say you're trying to find the highest peak in something that looks like this. And obviously there are multiple candidates. And so this might take a lot of searching, but with enough, we could be relatively certain that we found it. And we might get a set of answers that are very similar from that one and that one as far as what the highest peak is. A very different search topology would be something like this. And I picked Mount Fuji because no matter where you started in the surroundings around Mount Fuji, you would be going up the slope to find the very best overall one. And so if we have a very small set of phylogenies that are all in one kind of general vicinity with all very similar answers, it would be a fairly simple one. No matter where we started, we would end up recovering that. But with Markov chain Monte Carlo, we can also recover all the best answers, our population of best answers, even if our search topology is very, very rugged. And so we do random beginning points, and then we save all of the trees that are in this very good score area. We discard the phylogenies that have lower scores. And this is called the burn-in period. Now, during this burn-in period, and actually even during the plateau period, we also allow our model of evolution to vary. So let's do a step-by-step walkthrough, and I'll type it out here. But let's do a step-by-step walkthrough of what we do for a Bayesian analysis. Number one, we estimate our parameters for the model of evolution. And we do this, I'll put this here, it's a little bit long, but we do this based on our initial prior probability. Now remember, we don't know anything at all about the search topology. We don't know if it's Mount Fuji. We don't know if it's Superstition Mountains. We have no idea. And so our best estimate at the beginning, let's make that. So our neighbor joining tree, although it's not probably the very best overall set of relationships, it gets us in the ballpark. And that helps us then estimate our parameters for our model of evolution. And then we run some iterations. We use Markov chain Monte Carlo. We use that model of evolution with the parameters estimated from it to get an answer. And then we pause. So number two, run some analyses. And we'll put MC, this Markov chain Monte Carlo, as we run those analyses. And then at that point, we pause and re-estimate our parameters. So maybe as we're running this analysis, our scores for our trees, parameters, are getting a little bit better. And that's great. If they're getting a little bit better, that means our estimation of phylogeny is probably better. And then we can re-estimate our parameters. And then we repeat. And as long as our scores are getting better and better and better, we continue to do that. Then at some point, our scores will reach kind of this plateau phase where no matter how many times we go through these steps of repeating, re-estimating our parameters for our model of evolution, getting the tree scores out of it, we get this burn-in, or the burn-in period is gone, right? So that says trees are getting better and better and better. We reject all of those trees, and then we start saving all these trees that have really good scores. And many of these trees might be repetitions of ones we found before. We're finding the trees over and over and over again, but we're kind of dialing in our model of evolution. And then we repeat the whole thing, usually 4 times, sometimes 6 or 8 times. Repeat the entire process we'll say 4 to 8 times. You can do more, but after a while, you're kind of getting limited returns for your efforts. So we repeat this entire process 4 to 8 times. And each of these times when we start it, we're doing these random starts, we save all of these trees. So the end result of this is this large trees, hundreds of thousands of trees that we've saved that are all pretty good scores as we've looked at our entire search topology. We then make a consensus tree for all of these trees that we've found. So basically we compare all of the trees, we look at what relationships they have in common and what relationships are different, and we put numbers on those. And these numbers become our support value. So for instance, if this relationship between Pleodorina and Chlamydomonas, these 2 species, if that is found in every single one of those good trees that we've saved, then we put 100 there, representing 100% of the time. If a set of relationships, so these 3 species together, is only found there in 82% of the trees, then we put that number there. And if a set of relationships is not found in a majority of the trees, we collapse that node down and we make it a polytomy. So the nice thing about the Bayesian analysis is that we end up with support values as part of the process of doing the tree. We'll look at other support values in our next unit that are used for other analyses, but a maximum likelihood analysis gives you a tree, but it doesn't tell you if there are some of those relationships that are better supported than others. A parsimony analysis, the same thing. You get an end result and says this is the best tree overall based on this parsimony criteria, but it doesn't tell you if some of those relationships are better supported than others. A Bayesian analysis does that. So it's another time-saving part of a Bayesian analysis. So not only are Bayesian analyses overall faster than maximum likelihood analyses, but they also give us a support measure at the end of the analysis. So that's a huge advantage for Bayesian analysis. Okay, so review of some things we've talked about already. We can use known phylogenies like from bacteria and determine whether parsimony, maximum likelihood, or Bayesian analyses are best. And we can also use simulated data. And we've talked about areas like long branch attraction where parsimony fails. We talked about areas like rate heterogeneity, also called heterochrony, where maximum likelihood might fail. But overall, if we're in kind of a majority of these trees, we get pretty good results from even neighbor joining, but certainly maximum likelihood, parsimony, and Bayesian analyses are going to give us good results. But because of statistical considerations, many people want to use either maximum likelihood analysis, but that can be computationally intense. And so Bayesian analyses is a little bit of a less computationally intense way to analyze. And then we can look at results. So here's a result where different methods, weighted parsimony versus parsimony, find the very best tree. And notice that in most considerations, given enough data, we're going to find the very best tree. But some methods perform better overall, like maximum likelihood, even neighbor joining, if we have correct models of evolution. Okay, so the take-home message, again, is that we may want to use a variety of methods. If we want to use statistical approaches, we might do a maximum likelihood unless it's too computationally intense for the size of our data. And then we might want to do Bayesian, but also compare it to a parsimony analysis. And our best overall phylogeny might represent a consensus among methods. And so this congruence among methods is a very good way. And if we have areas that are not congruent, then maybe we want to reevaluate or gather more data to determine if we can resolve that. Or maybe we say, well, I'm going to use the maximum likelihood result or the Bayesian result because I want to do a falsifiability test. I want to use those statistical approaches to be able to do that. And so here's our big picture overview of all of the methods that we've talked about. Now, originally, I put the Bayesian analysis in this category of using nucleotide sites, and we certainly do that. We're not using distance data, so it's very clearly in this right-hand column. But whether it goes in a clustering algorithm or an optimality criterion is a little bit different. In both parsimony and maximum likelihood, we really are optimizing. We're doing this by doing a very intensive search of lots and lots of trees, and we're really optimizing the score based on either a parsimony criterion or this likelihood score that we gave you an overview of. In a Bayesian analysis, we're doing something slightly different, which is why I've put it up here, but with a question mark. We are really optimizing the model of evolution and the parameters for that model by doing this iterative approach, where we start with an unknown distribution. Maybe we give a neighbor joining tree. We then allow the analysis as we progress to inform it and to then go back in and re-evaluate or change the parameters for our model of evolution and then see if that gives us a better tree. If it does, great, we keep them. If not, then we allow it the next round to change those parameters for the model of evolution. So in reality, a Bayesian analysis is optimizing our model of evolution and our parameters for that model of evolution, and then allowing that to inform our overall outcome. So it's not really, maybe a better place for Bayesian analysis is kind of in the gray area between these two things, because we're not really finding the best tree based on a single optimality criteria. We're allowing our analyses to inform the parameters, change our model of evolution, or at least the parameters for that model of evolution, and then reiterate and do it again and again and again until we don't get any better. And we've kind of reached a plateau phase where we're not getting better scores no matter how many times we do this iterative approach. So review this, make sure you're familiar with these, albeit somewhat of an overview of methods for some of these. You should know how to map characters onto a tree in a parsimony analysis and find a score for that tree. And then just understand the basic principles for these more computationally intense methods, maximum likelihood and statistical methods for Bayesian analysis.